Okay, so this is the third method we're talking about. Well, really fourth if you count the ribo as some hybrid. Okay, so how does nucleotide excision repair work? Okay, so the first thing to know is that this acts at sites of bulky DNA modifications. Okay, so the best example of this would be a pyrimidine dimer. Okay, so this is our second method to attack pyrimidine dimers, but other modifications that you see in DNA that dramatically distort the DNA will induce nucleotide excision repair. So for example, if you have an A across from a G, that kind of mismatch causes a bulky part in the DNA, and that's also recognized. And there are many modifications of DNA that cause it to be bulky as well. Okay, so we're going to start by talking about the E. coli system. Okay, and there are 4 proteins required for this called UVRA, B, C, and D. Okay, and fortunately, not only are they named that way, but they're actually used that way, which makes life a little bit easier. So how does this operate? What's it recognized to begin with? Well, it recognizes bulky DNA. It's not as easy as the mismatch where they have a crystal structure and they can really understand that it's some ability to distort the DNA that's recognized. But like the mismatch repair, this stuff is going around all the time and scanning the DNA. Okay? So these proteins, UVRA and B, which form a A2B2 complex, okay, scan the DNA for bulky adducts. So not entirely sure how the scanning goes. We know less about the on rates and off rates, et cetera. All they know is that when you have normal DNA, it binds and releases relatively quickly. Okay? If it's okay, but the more relevant situation here is if they detect a lesion, then 2 things happen. Okay, first, binding is enhanced, and second, UVRA hydrolyzes its bound ATP, and that causes release of UVRA ADP. Okay, so the ADP form of UVRA does not bind to UVRB well. Okay. So it binds tightly when it has ATP bound. It binds weakly when it has ADP bound. Okay, so that leaves UVRB behind. And UVRB does the first important trick of this process, which is that it now can recognize which strand has the modification. Okay, so UVRB remains on the DNA. Okay. It then melts the DNA. It has a helicase-like activity. Okay. Okay, and then its really nifty trick is that somehow, and again, no crystal structures, so we don't know how, UVRB is released from, make sure I get this right, the damaged strand. Okay, so the simplest way to think about this is it likes to bind normal single-stranded DNA and not damaged single-stranded DNA. But the part that makes this a little bit more tricky to understand is if it's really recognizing multiple base pairs of single-stranded DNA, how does having one damaged base really change its affinity that much? Okay, but that's awaiting things like crystal structures to make sense of it, I think. Okay, right. So the next step is that the UVRB that remains behind binds to a molecule called UVRC. Okay, and UVRC probably is the most important of these. This actually has the ability to cut the DNA at very specific positions. So this is an endonuclease. Okay, and it cuts 5' to the damaged base. Actually, write this up. Cuts 8 base pairs, 5' from the damaged base. And 4 to 5. 4 to 5 base pairs, 3' from the damaged base. Okay, so it nicks on both sides of the damaged site. Okay, and B is what's sending it the signal as to which of the single strands it acts on. Okay, because B is still bound to the good strand, it's released from the bad strand, and it's the bad strand that it needs to cut. Okay, so it's actually a dimeric multi-endonuclease protein, right? It's actually, I shouldn't say it's dimeric. It has 2 endonucleases in it. Okay, so it's a single UVRC, but it has 2 places it cuts, and it's somehow measuring, okay, from this damaged site to cut in exactly, or at least on one side exactly, and on the other side within 1 base pair of a particular distance. So again, it's recognizing the damaged base, but it's not clear how it's recognizing the damaged base. It might be that it's just looking at, for example, just for some sort of bulky position, but it's a little hard to understand. I mean, this is particularly tricky when you have a thymidine dimer because you're sort of looking at 2 bases. Right? But this works very well on thymidine dimers. In that case, it's the 8 base pairs is upstream on one side. The downstream, the 4 to 5 base pairs would be on the other, measured from the other of the pyrimidine dimers. Okay, so now we have what we really want, and it's pretty much easy going from here on out. The only thing that helps it is the last of the UVR proteins, so UVRD, which is also known as helicase 2. It's another name for it, removes the, let's call it the excised single-stranded DNA, and then that is simply acted on by DNA polymerase plus ligase to replace the damaged DNA. Okay? So that's what's really illustrated here. The UVRD molecule just peels this strand off, and then DNA polymerase and ligase resynthesizes across there and seals the nick. Okay? So you've basically replaced very specifically a 12 to 13 base region that was damaged before and is now, hopefully, as long as the polymerase did its job right, replaced with wild-type DNA. Okay. Any questions about how that works? I mean, other than the questions that we have overall, which is it's not really clear how the damaged DNA is detected, unlike the situation with MutS, where we really have a very good molecular understanding of that. Okay, yeah?
What is it that you mean by if B-type DNA released quickly?
So if it's scanning all the time. So B-type is normal structure of DNA, no bulky adducts. So if it's just scanning along and it sees, oh, this DNA is normal, it just falls right back off. It's only when it detects some distortion in the DNA that it stops and starts well, first of all, the A is hydrolyzed, then B can start to melt the DNA, and presumably that's the point at which you get strand discrimination, right? In the end, you don't want to replace both strands, you want to replace the damaged strand, and B is performing that magic trick somehow, right? It's the thing that can say, the strand I'm bound to is fine, the strand I'm not bound to is not fine, and bring in C to cut the strand it's not bound to, okay?